Hallo zusammen und guten Abend miteinander. Ich hoffe, es kann mich jeder verstehen. Ich begrüße euch ganz herzlich zum heutigen Abend in der Shark Awareness Week von SHARKPROJECT, unserer Webinarreihe. Mein Name ist Anke Arbeit, ich bin von SHARKPROJECT Switzerland. Ich mache eine kurze Einführung in Deutsch und würde dann auf Englisch übergeben an meine Co-Hosts, die Eleanor Spencer und den Gibbs Kuguru. Wir haben heute das Webinar, die Eleanor wird berichten zu unserem Austausch und unserer Zusammenarbeit mit dem Greg O'Connell. Da haben wir verschiedene Projekte, die wir miteinander bearbeiten und wo wir aktiv sind, und sie wird über die Ergebnisse berichten, Projekte, die wir von SHARKPROJECT aus unterstützen. Und Gibbs wird dann weiterhin fortfahren, welche Auswirkungen ein fehlender Umweltschutz haben wird, was die Auswirkungen auf unsere Natur sind und was hat das mit Leuzismus bei Haien insbesondere zu tun. Er wird auch noch den Bogen schlagen, warum ist es wichtig, dass wir gesunde Haipopulationen haben und wie wir diese erhalten können. Da gibt es viel zu erläutern. Es ist ein sehr breites Themengebiet, deswegen möchte ich dann auch nicht so lange warten. Ich würde euch bitten, stellt eure Fragen über das Q&A, Fragen und Antworten. Wir haben die Funktion Handheben im Moment außer Kraft gesetzt. Deswegen bitte ich euch, das über Fragen und Antworten eure Fragen zu stellen. Wir werden diese so gut wie möglich schon während der Präsentation beantworten und sonst nachher noch. Insgesamt wird unser Webinar circa 1 Stunde dauern. Ich wünsche euch ganz viel Spaß und sehr einen interessanten Abend. Und ja, let's go, Eleanor. Dann würde ich jetzt noch schnell die Eleanor vorstellen, auch dass ihr wisst natürlich, mit wem ihr es zu tun habt. Sie hat einen Master in Meeresbiologie der Universität von Southampton. Sie ist Mitglied von SHARKPROJECT Switzerland und sie ist Projektkoordinatorin mit dem Greg O'Connell, und in dieser Funktion ist sie heute Abend anwesend.
Hi. So today I'll be going through the project that SHARKPROJECT Switzerland funds with Craig O'Connell, known as the White Shark Nursery project. This is Dr. Craig O'Connell, and his team have been working with SHARKPROJECT Switzerland on this project for the last few years. Because it is Shark Week, I'd like to introduce you to him, he himself is a Shark Week presenter, and he's been doing actually around 3 different documentaries over the last week for different shark-related projects. Just a quick factoid about if you are interested in Star Trek or anything related to that, he actually has recently, around 3 days ago, I think it was, he actually did some work with William Shatner and some other famous faces. So if you are interested in that sort of area at all, or a Trekkie in any sense, I'd highly recommend checking him out. And you can go to his Instagram. I think his handle is known as the Shark Doctor, and it's really interesting stuff that he produces. But to bring it back to the project itself, Dr. Craig O'Connell, he is both the director and founder of the O'Seas Conservation Foundation, which mainly focuses on shark research and youth conservation education camps, which takes grades 8 to 12 out, and actually allows them to do this research with them, which includes the work that is done on the White Shark Nurseries. This isn't the only project that we actually helped to fund him with. There are 3 projects in total. So this includes the White Shark Nursery project, another study for capture stress, looking at how capture can affect sharks, And a third one, which is our most recent one, is looking into barriers that are safe for sharks as well as for humans. But this project is quite new, so we are still waiting for some more information about any of the findings from these projects. So let's discuss what is the White Shark Nursery project then. This has been a project that we have been running with Dr. Craig O'Connell for 4 years now. Since 2017, we've actually been funding this project. This project actually began when Dr. Craig O'Connell and his team had actually started noticing, actually been alarmed by the amount of juvenile white sharks they were actually seeing being found in fishermen's gill nets just off the coast of Montauk, New York, which was actually quite surprising because at the time they were unsure if there were actually people didn't know that there were actually white shark nurseries out there. So this actually started making them start to question why this was beginning to happen, and so therefore this project began. The main goals for this project is to propose specific protection measures for when white sharks are in the Montauk area, but to actually take effect precisely when the white sharks are in the nursery. So not to actually take effect when they're not there, but only when they come around, because, well, with white shark nurseries, it's only a particular time of year that they're actually going to be there. And also this is also to prevent anything impeding on fishermen's in certain time of year where you don't always need those protections on when they're actually not there. The next one is to collect more information on the common sharks. So the reason for this is actually certain shark species are actually highly representative in the diet of white sharks. And third one is to implement protective measures for a variety of shark species. Because, well, if you protect the prey of the white shark, you protect then the white shark itself. This is known in marine biology as the bottom-up approach, meaning that by protecting the lower levels of the tree, you're actually helping to protect the higher up levels as well. So, what is a white shark? What is a white shark nursery? There are actually 3 criteria we need to pass in order to determine what a white shark nursery is. So first one is there needs to be white shark neonates. These are juveniles and newborns that have to be sighted within the area. The second is that these sharks need to stay or return to this area over extended period. So this needs to be actual specific individuals need to return to the site within a year. And the third one is that the sharks need to utilize this habitat across years. So not the same sharks, but newborns and juveniles of the same species need to use this habitat year after year. So the next big thing, and this is more of the technical sided stuff, so I will try to take my time and not just try to speak too quickly. So how do actually Craig O'Connell and his team help to identify what a white shark nursery is? Well, what they need to use is a specific technology known as BRUVs, which are baited remote underwater video systems. These are a completely non-invasive technology, so there's no capture involved, it is just filming. So they actually work in a similar way to how game cameras work, is that all they're doing is they're just recording the species and trying to see what's there. They don't work by actual motion capture like game cameras. How they work is they run the entire period. So once they're put into the water, they record these particular ones that Craig O'Connell uses, actually record for up to 16 hours at a time. And they were actually usually used between the months of May and November, which is the time period when we know that shark mothers will be actually releasing their eggs and juveniles, newborns will be around. So how do these BRUVs work? Well, the main thing that you need is a camera and a bait, which is usually always included in a bag. So that's probably the most basic form of what a BRUV is. And then the camera will just record what comes by to the bait or what is attracted to it. But with Craig O'Connell's system, it actually with different species, you have to have different rigs set up and you have to have more complex systems. So with the bruvs that Craig O'Connell and his team use look like these. So the reason why you have this big squared off area here in this yellow box is because actually this is used to actually counterweight the bait. So then that actually we get more stable footage of when actually the bruv is being put down at the seabed. So then you're not getting too much movement and it's got some stabilization. You will always have the bait attached here at the end, if I don't know if you can see this, right here at the end of these like hooks where the kids' hands are, as you can see. Then what you'll have, you always have to have attached is a red buoy, which will actually go up to the surface and will be attached to this point. And what we'll be doing is that it'll actually indicate to both fishermen and also to the scientists where they've actually put the bruv so they can help to re-find it after the 16 hours of waiting. But then this part here actually is probably the most interesting, is that you've got this bar here in the middle, which is for the GoPro camera to be mounted. And then you have this other additional one known as a decoy bar. So the reason why you have a decoy bar put into these systems, or why Craig O'Connell has actually put them into this system, is because what they found was the sharks were actually attacking the bruvs, or more likely were actually biting the GoPro cameras. And the reason why you have a decoy bar is actually to prevent the sharks going for the cameras. And actually, so they'll be biting the bar instead of going for the camera. And the reason why actually sharks do this is because that, unlike humans, they don't have hands. And sharks are actually a very curious species. And because they've got so many receptors and so many, like, in their mouths, they're actually really sensitive to certain things. What they'll do is they'll actually bite, they'll actually out of interest, because if they miss, or if they're becoming too rough with the bait bags, they might accidentally bite the camera or ruin the footage that have been taken so long to record. And that could mean that the scientists have lost all that information. So a decoy bar helps actually prevent them, like helps to like redirect their curiosity in a sense. So it's not, when they actually release the bruvs, this is not the only information they collect. So when they're actually recording the species, they actually can identify individuals when they come up to the cameras based on the markings on the dorsal fins. So the top fin, the biggest fin you get on the shark, that's how actually scientists can help identify what individuals are coming by. They also try to look into the individual species because, as you know from one of their goals, they're trying to identify the different common sharks that are within the area, as well as in some cases they can identify the sex of the shark based on their claspers or based on like how their undersides look, because they have like different looking parts there. But then in addition to both the recording stuff, what they also try to collect is, once they've released the bruv, because there needs to be more holistic outlook of like the area of what they're getting, is they both collect the information of like what the temperature is of the water depth, the moon, as well as the tide cycle, because one, they want to understand where and when juvenile white sharks are found, as well as trying to identify how the environment and the biology may or may not influence the presence of these sharks, because, well, anything with biology speaking, you have to have a holistic picture. So all of this information is required and needed to understand why and where white shark nurseries appear. So with using all of this technology and all of this information they've collected, what has been so far out of the last 4 years been the outcome of this big project? Well, they have actually identified 2 white shark nurseries off the coast of Montauk, New York, which is a big thing for them as well. The findings from their studies have been published in the Marine Technology Society Journal in February of this year. In addition to this information, both included their developments with their bruv systems, as well as the identification of different nurseries habitats they found off Montauk. And finally, and this is in the world of science and in conservation, this is like quite profound to have happened. In May of this year, the region around Montauk has been upgraded to a high level of priority protection by the New York Environmental Protection Agency. So what so from this, what they've actually really wanted, what this means is actually this area or the heritage site will actually provide greater protection for this Montauk region. But in addition to this, what the New York Heritage Program have asked is to collect the data from this project and other projects in order to create a systematic monitoring study in order to identify when great white sharks are in this area. So this can then influence both how they enlist the protections of the area, how they inform people where they can go fishing. For like in the world of conservation and in the world of like science, it's very rare to actually have your science being used and all your findings actually being used for real world applications. So this is like, considering that this project has been 4 years, it's really profound that this has happened and it's incredible. But within light of all this amazing news, there isn't always that plain sailing. So things can mess up during this project as well. So for the first, actually in 2017, when this project first began, actually 10 bruvs were stolen by potentially fishermen or sports fishermen or whomever, just because, well, I think this is an important point to bring forth as well. For any fisherman, you shouldn't really think that, oh, this is like they're being horrible, this is all completely wrong. And yes, in the science of science, it is. And it can be really horrible to have to go back over and over your data. But the understanding you have to make for when fishermen do this is because, well, if you enlist a protected area in a certain area where they go fishing normally, this can actually really hinder their income they've had for years, and it can be really profound on them, especially as if it's never had an issue before with like sharks. So I think one thing that Craig O'Connell and his team are really trying to do is both create a communication channel between themselves and the fishermen, which has really helped over the last few years, and no more theft has occurred and anything like that. But also it's to provide better education, I think, both between the fishermen, but also between the conservationists, because I think for ourselves as conservationists, we need to understand where fishermen are coming from and how we can all work better together. So I think that's a positive that has happened from this incident. And obviously, with everything going on, you always have to talk about the COVID situation. So actually, there has been a lot of limitations that have been caused to the research. Because there's been so much limitations, this funding was meant to end last year in 2020. But because of this, we've actually agreed to another extension in order to help Craig O'Connell further extend his project, because one, he actually came down with COVID-19 and meant that he wasn't able to dive as much, and he actually had to withhold a lot of his study for an extended period. And also because of the whole situation, his camps that usually help to subsidize some of the funding were completely taken. But then now, luckily, with everything opening up on that side, in America anyway, they've actually started to come back and bring back some of their income. But again, with everything, it's just slowed some things down. But there are some signs of recovery, which is good. But in light of everything, it's good to mention that there have been limitations. So thank you so much for listening. If you have any questions, I'd love to answer them.
Thanks a lot, Eleanor, for your speech and a lot of information about our project with Craig O'Connell. Please don't hesitate to ask Eleanor your questions. As additional information, the cooperation with Craig O'Connell, it's going forward. It isn't stopped. So we have a new project with him. We are working together, and he is working on other projects. So you will find him on YouTube on different channels. And as Eleanor said, you will find him as well during Shark Week in different documentaries, and such an interesting cooperation. Any questions? Maybe Eleanor, you can tell us about the other collaboration with Greg, the new one. Maybe it's interesting for our audience.
I think, well, there's like 2 current collaborations, and I think one of the most interesting ones I've got the most information from Craig about is our capture stress study, which actually is looking into how sports fishing could actually influence, sorry, I should actually say that again. How actually, because if you do sports fishing, you know that the whole purpose of it is that you capture the fish and then you re-release them after a period. But what will happen is that with certain types of shark species, that if you capture them and then re-release them, this actually doesn't mean that the species is actually going to survive this encounter, because certain shark species can actually be really sensitive to stress. And so what Dr. Craig O'Connell and his team are really trying to understand is how this capture and how certain species will react after a certain length of capture. So they're looking up to 5 different species, I think the white shark, the common thresher, and some others which are now eluding me, how they're trying to understand how each species will be influenced, because some species after say 20 minutes of capture or far less will actually be completely fine after an hour and days, and will last days after the capture and be completely fine. But then others are highly sensitive, and after 20 minutes they will die once they're back in the water after a few hours. So what they're really trying to do with this particular study is try to actually provide some information to sports fishing regulation and things like this to help influence people in the sense of saying, well, if you capture this particular species, make sure to re-release them straight away, or you can handle these ones, or actually to just completely throw away or completely say to people, you just cannot capture certain species because it just will not survive any sort of capture. So it's a really important study in that sense, because obviously with sports fishing, you can't really completely stop it. Though we wouldn't want it always, you can't stop it. So they're trying to find ways of helping to protect species better. Then the newest project they have, which is looking into sharks, is safe barriers for sharks, in the sense of trying to prevent sharks going into certain areas by creating barriers that aren't going to harm them or anything. They're just simple systems, but they're trying to reduce the amount coming in. This is also trying to help protect swimmers in certain areas, but also mainly to prevent shark culling in certain regions. So you'll get in places such as, and I'm trying to remember the location, so I apologize if I get this wrong, which is I think in Bimini or Bahamas. There's some speculation that they're actually going, because there are so many sharks in certain areas and they don't want them to interact with swimmers or divers, they are considering doing mass culling events of these sharks. And again, if I've got the locations wrong, I sincerely apologize. Then what they're trying to do with these barriers is by creating exclusion zones, so that then the sharks won't come into certain areas, and actually then these culling events won't be needed. And so that's why his research he's currently doing is so vastly important and really influential. That's the main thing of what the current projects are up to.
Thanks a lot, Eleanor. So, any questions yet? Otherwise, we will switch to Gibbs, to his studies. And yeah, we are working with Gibbs since I think 2 years. I met him in South Africa, and now we are partners in Shark Project. Shark Project Switzerland supports his PhD research in leucism in blacktip reef sharks. Yeah, Gibbs, he's working and studying sharks for years, and when he isn't in the laboratory for research work, he's in water, underwater, to dive with sharks, to work with sharks. And I think we will see in the following years lots of him at the TV. He's working with National Geographic and in different documentaries with research in sharks. So I have it's enough introduction of you, and maybe you can tell something about you too. So yeah, it's your turn, and yeah, we are interested in your presentation. Thank you.
Thanks for the introduction, Anke. Can you hear me okay? All right. Let's just see if I can quickly share my screen. All righty. Are you guys able to see my presentation? Super. Thank you for the introduction again, Anke. Indeed, my name is Gibbs, and I am a shark researcher. I study the DNA of sharks, so I technically would be called a shark geneticist. But moreover, I think it's most important that I do identify also as a conservationist. And when I talk to other ecologists or shark scientists about conservation, I'm quickly alerted to the fact that I have kind of a different perspective about conservation than they do. And I think it's important to identify what causes this difference in perspective. And I think that's primarily because traditionally conservation looks at how many animals or how big a population is based on the numbers of individuals are left. But I study DNA, so I tend to look at the molecular level and how threats facing wild animals actually impact the DNA of the populations. And this is something where a traditional conservationist would look at numbers. I would look specifically at biodiversity and why conservation matters to preserve biodiversity. So in order to really describe what's going on there, we really should start from the beginning, which starts with the DNA molecule. This is a 3D rendering of the DNA molecule. And I don't want to get into the structure of it, but I just want you to take a look at it and just imagine that this single molecule is responsible for every living thing on the planet. Every tree has a piece of DNA that controls it. Every fish has one. Every bacteria, every virus has a piece of this in it. And it has the ability to code for a wide range of adaptations. We have seen animals present the ability to fly, to camouflage or disappear altogether, and even to suffer and withstand the most severe traumas. And it's all housed in this very simple molecule that we all share with every living animal and plant. And when most people think of geneticists, you're often reminded of movies that you've seen where you have a scientist standing in front of this really beautiful holographic computer, and with the flick of their wrist, they're able to just change the DNA of a certain species and modify it, and really, it really looks like cool science. But actually, what it mostly looks like is you're sitting in a lab, everything is fluorescently lit, and you're most of the time just transferring clear fluids from one test tube to the other. And you do this for a considerable part of your career. And this is how we sort of learn to manipulate our DNA. And while it looks like this, after a few months of doing it, it starts to feel a little bit like this, where you're sort of sitting in your lab for hours over a series of weeks, and you feel like there's no end to the amount of lab work you have to do until you produce your data. And our data looks like this. It's a series of DNA codes, and these codes tell us the instructions for each animal or plant or living thing that exists. And we now have really amazing computer science tools to analyze this, so we can really understand what's going on in nature. Though even with all of our technology, the amount we know about DNA and how it actually works is so little. So now that we're actually in this fourth industrial age of information technology, I think we'll be able to get a lot more really interesting facts about how DNA works and how it shapes our life and our environment. So yeah, genes or genetics is the study of how genes are basically passed from one generation to the next. So as a geneticist, our focus is really on the events prior to and subsequent to the event of sex. So without sex, one would say that a geneticist doesn't have a job, which I believe is true for very few jobs that exist out there. I'm actually working in a particular field of genetics called conservation genetics or conservation genomics, and we're primarily invested in using genetics to not only understand the populations we work with, but to actually see how we can improve them, because we see a stark loss of biodiversity across all of our animal and plant systems. And this is primarily caused by people. So I'm sure a lot of you shark enthusiasts have probably seen an infographic like this about how many sharks are killed every year by people. The numbers are alarming, and this is the primary driver for why sharks are under threat. It's simply because people the economies have grown and populations have grown, and so has the demand for food. So the more people you have, the more food you'll have to feed them. And we are also living in a culture of excess. So it's not enough to just have enough. You have to have more than what you actually need. And this is actually a problem with how we do things, and also affects sharks as well. So what happens then when these threats run amok and there's no real effective conservation? So let's rewind about 100 years. And let's just say that our oceans are full of sharks, a sea full of very large and healthy animals, and they're just going about on their merry way. Now, whilst a traditional conservationist would see each individual shark as a specimen, I, as a geneticist, would actually look at them as the genetic code that actually encodes for that animal. These little genes actually host all the information for the animal, and it's vital that they are able to pass that on to the next generation. So in a sense, I would actually take a look at that sea of sharks and break it down into their single genetic units, or DNA molecules. And well, from this photo, you can see that it's kind of in a ratio. I've separated them based on their fitness. This is an arbitrary measure, so I'm just saying fitness is the ability for an animal to withstand a certain amount of trauma, for example. So let's say we have about 25% of the animals being super fit. I hope you can see my mouse. About 50% of them seem like semi-fit, they can handle themselves. And then at the very bottom, we have another 25% of these sharks that seem to be a little bit unfit, i.e. they need all the help they can get. And this is in genetics what we call equilibrium. You can see that the sharks have found a way to balance their populations so that they can not only maintain biodiversity, but also maintain the health of their populations. And what I'm going to do now is introduce a series of stresses or pressures onto this population to see how these sharks will actually adapt to it. So the first stress is going to be anthropogenic stress, which it just means people-based stress. Then we'll introduce an environmental stress, and then lastly, a biological one. So we have our super healthy population here, and the first stress we're going to give them is the people-based stress, and that is in the form of overfishing. As I said before, in the last 100 years, people's economic status has allowed them to purchase more food than they need, and more often than not, there's so much food waste, like what's happening in this photo. The shark has been finned, more or less, has been the ability for that shark to swim has been taken away for it, so it just sinks to the bottom and dies. And this happened for quite a number of years in our seas. But so we have these genetic units of these sharks. They seem to be doing quite well. And then we start our first round of fishing. And we take out a whole bunch of sharks. And if we just left the population as it was, it should have been fine. Because you can still see that the equilibrium in the population still seems to be quite stable. But that's not what people do. We go for the excess. We want to get more, we want to get bigger, we want to get faster. So we keep fishing. And a series of fishing events later, then we can see, okay, our population is a little bit different. You can see that it's a little bit out of balance. And this, for example, in the 80s, this started happening where people started seeing, conservationists and scientists, started seeing all these sharks coming out of the ocean. And they said, guys, I don't think the ocean can handle this much stress. And, you know, a lot of these industrial complexes would say, well, we've actually had our catches remain steady, and we're even able to catch them more than we've had from the 50s and the 60s. So surely the ocean is full of sharks. We haven't even scratched the surface. And of course, without proper government regulation, the fishing continues. So another round of fishing goes, and now your population has dwindled. And on the right, these ratios represent what it started as the equilibrium, the balance. And on the left is the more or less after photo. And you can see that there is a slight difference in the population. The really fit, the strong individuals in the population are less. And the semi-fit and unfit ones are a little bit more. And well, in a perfect world, I guess nothing would be wrong, but we don't live in a perfect world. And then we get to our environmental stress. So, well, you can see that the populations now have sort of, the numbers have recovered. So now the traditional conservationists would say, yep, we're really happy. The sharks came back. But yeah, our ratios are different. And now we introduce the environmental stress. Disease. Let's just say randomly that there's some virus that gets randomly introduced into the ocean and starts infecting a whole bunch of sharks. And well, let's just see what happens. So let's just say that this disease targets this population of sharks. And now you have one of your fit sharks quite sick here. About half of your unfit sharks or semi-fit sharks are plagued by this disease, and then all of your unfit sharks in red succumb to this disease. So now your populations look a little bit different. And you can also see that now that the numbers are also lower, the genetic diversity and the biodiversity is gone. Well, you've lost a whole genetic unit of these unfit individuals. And now your ratios look like this. So as you see on the right, you have the before photo in equilibrium. And on the left, you have the sharks that, well, represent what's happening after these two stresses. And you're probably thinking, and I can tell, you're probably thinking that, oh, look, you have more fit individuals and more unfit, semi-fit individuals. Isn't this better? But actually, nature has told us otherwise. And there's a part of diversity. When we talk about diversity, well, when it comes to people, it often gets lost on what that actually means. But when we see it happening in nature, when you lose diversity, or when you over specialize into a certain type of thing, whether it's a fitness or large size, for example, eventually you breed in weakness, because diversity is a strength of a population where over specialization is the weakness. So we have a case where now our breeding individuals are closely related, and we get into a scenario of inbreeding. And I hope this photo wasn't lost on you. For those of you who haven't seen Game of Thrones, these two individuals are siblings, and it's a way of representing inbreeding for me. But more or less what's happening is, with inbreeding, closely related individuals are not able to select other mates. So they end up mating with the ones that are closely related to them, which is a problem, especially in nature. So we have our super fit sharks and our semi-fit sharks, and they're all getting closely related because there's few of them, and they more or less have to mate with each other. And well, inbreeding is not without its consequences. So a few of these sharks are now going to succumb to the negative effects of inbreeding. And well, from what we've seen all across nature, is there are things like congenital birth defects, growth deformities, smaller size in adulthood, and even a higher incidence of cancer that actually prevails throughout these populations whenever you have a high degree of inbreeding. And yeah, you're probably thinking, well, where has this shown in nature? How have we seen this even in sharks? And I'd like to now talk a little bit about my PhD research. So a couple of years ago, I saw this exact photo of these sharks in the Maldives that sort of, it looked peculiar, but I just took a glance at it and I thought, oh, what could it possibly be that causes this weird thing? Nothing bad, right? It's just a patchy nature to their skin. And I sort of put the publication away, and I eventually revisited it and started looking into what causes these spots. And I started reading about this disease called leucism, which these sharks have. It's actually a genetic disorder, so it's inherited from these inbreeding-related stresses. And leucism happens throughout the animal kingdom. It even happens in humans. Some people also know it as the condition called vitiligo, but it also comes in the form of another disorder, which is a bit more serious, called Waardenburg syndrome. And this comes not only with just a weird skin pattern, but it's actually a neurological disease that affects any part of your body that has neurons in it, or nerves. In horses, for example, leucism can cause blindness. And in dogs, a leucistic dog can actually have deafness. And we've seen throughout the animal kingdom things like an immunity that's compromised, so the animal isn't able to sort of fight off diseases. And the scary thing for our sharks, these sharks in the Maldives, is they've only been seen with this disease when they're in their juvenile, or when they're young, life stage, which means that, well, there's two scenarios. The first being the best case scenario, that these animals are more or less growing out of this disease or this genetic disorder. The less positive one, and the one I think is likely happening, is that they are succumbing to early onset death, i.e. juvenile mortality. And that's the reason why we're not seeing these sharks as adults. It's well, scientifically, it's terribly exciting. It's one of the most unique problems that I've ever faced as a scientist. But as a conservationist, I must say, it's alarming, and it's terrifying that this is happening. So how do we fix this? I suppose you could also ask the question about how do you fix a broken stained glass window that's been completely shattered? And the answer is it's probably better not to break it in the first place, because fixing it is it's not something we know how to do yet. I don't want to sound all doom and gloom, but my goal here is to work with organizations like Shark Project Switzerland that try to promote conservation measures that help give these animals the best possible chance to survive by changing how we do things, how we get our food, how we use our electricity, and how much we burn fuel. These are the things that might help change our planet, because trying to tinker with the DNA or do other things, whilst it is an option, it's not the perfect option. And even with all of our information technology and all of our understanding about nature, I think we really don't know how to do this. But it's my goal to make sure that we make it as amazing as possible for these animals in the future. So yeah, that's all I got. So thank you guys, Shark Project, for supporting my research, and cheers to everyone for coming to this talk.
Thanks a lot, Gibbs, for your information. And yeah, for me as a non-scientist, is every single time it's so educational to listen to your speeches. And yeah, thank you.
Cheers.
So any questions by our audience? Feel free to ask.
You can put your questions directly in the Q&A or chat, or if you have the ability to use your microphone, you may also ask that way. No questions. I suppose it was all clear.
Or maybe it's too much information. At the moment, and it's everybody's overwhelmed by this. Yeah. If there aren't any questions, we recorded our webinar, and you will find this on our YouTube channel on Shark Project International. Oh, yep. There's a response.
Oh,
and
cheers. Thank you, Robert.
And otherwise, you can follow our blog on sharkproject.org. There are a few information by Gibbs too about this project, and you will find this on our blog and on our new website. It's in progress. So thanks a lot to Eleanor and Gibbs. You can think about your questions. Otherwise, Dankeschön for your interest in our webinar. And thanks a lot for your insights in that interested projects, and we are able to support and that we can cooperate in this. Thanks a lot, and kurz Nächtle und ciao.
Yeah, good night, everyone.
Thank you very much.